Alrighty, so let's go ahead and get started. Hello, everyone. My name is Dr. Elizabeth Travers, and welcome to the Global Viruses Network Frontiers in Virology webinar series. And today I'm very pleased to introduce our speaker, Dr. Rajesh Kumar. Dr. Kumar works on developing immunotherapies against viral antigens. His lab has been developing in-house naive universal phage display scFv and nanobody libraries that can be potentially used to isolate antibodies against diverse target antigens. Additionally, the library's ability to select binders with nanomolar affinity from a variety of targets offers substantial advantages for applications in drug discovery, diagnostic assays, and therapeutic development. His works on developing biotechnology innovation platforms focus on accelerating antibody discovery using high diversity nanobody libraries, phage display screening, and advanced antibody engineering technologies. Dr. Kumar is also director of AbSure Biosciences, which focuses on developing next generation biologics for therapeutics, diagnostics, and research. Dr. Kumar, welcome and go ahead and take us away.
Yeah, thank you, Ruby, for the warm introduction. So let me start my presentation. I hope my screen is visible.
It is.
Yeah, yeah. So thank you. My talk is antibody-based therapeutic intervention for the viral infection. So I basically worked in Department of Bioscience and Bioengineering, Indian Institute of Technology, Roorkee, India, and you can see the beautiful picture of our campus. So it's the oldest institution in India. So approximately 180 years old institution, and it's approximately 3 hour drive from the Delhi International Airport. So before joining this institute, I did my PhD on HIV viruses from Department of Biochemistry, All India Institute of Medical Science in New Delhi. And then I worked as a scientist and senior scientist in Translational Health Science and Technology Institute, Faridabad. And then I worked 1 year in Institute of Advanced Virology, Kerala. And then I joined as a assistant professor here in Department of Bioscience and Bioengineering as a virologist. And my lab mostly focus on developing these antibody-based therapeutic intervention against the viral infection. So the work I am going to discuss is on mostly the part that I have done in Translational Health Science and Technology Institute, and then the work that I'm recently doing in Indian Institute of Technology, Roorkee. So as we know, for any disease intervention, especially for the viral infections, there are three major counterparts. Either we can make vaccines, and these vaccines are the best options, and they can help us in preventing the viral infections. And second is we can make small molecular drugs, especially antiviral drugs, that mostly used as a therapeutic intervention, but they can also be used as a prophylactic intervention. And then recently, these antibody has also been explored in the case of the viral infections, but in case of cancer and autoimmune disease, they are used in therapy from the last 2 decades. But in the viral infection, they have been used from the last 5 to 10 years. And we know the antibodies against the RSV infections and recently approved for the Ebola, and one antibody has also been approved for the HIV, and few of them was also approved for the COVID. So these antibodies can be potentially used both as a therapeutic and the prophylactic options. So my lab works on developing these antibodies. So we make these antibodies in our lab, and then we have different viral assay systems in our lab. We look for the neutralization potential, binding affinities, and then where these antibodies bound, especially for the epitope mapping. And then we finally go for the structural studies in collaboration with the X-ray crystallographer or the cryo-EM people. And these are some of the picture of the nice HIV trimers that we have done in one of our studies. So we mostly work on developing these antibodies by the phage display technology. So we have developed our in-house naive human antibody libraries, and we have nanobody libraries from Camel and Sharp. So We work on different aspects of the viruses like COVID virus, chikungunya virus, and some of the flaviviruses. So once these antibodies are there, we test them for their biochemical and biophysical properties, and then finally we test them for the neutralization potential, whether these antibodies are able to neutralize or not. And then finally, the lead candidates are finally tested in the animal models for their prophylactic and therapeutic applications. So we know these antibodies can be generated by three different technology platforms. The first one is hybridoma, second one is the recombinant technology, and the third one is the single memory B cell technology. And other technologies are also like transgenic humanized mice are also used for developing these antibodies. So my lab mostly works around the recombinant technology and especially against the phage display antibody based technology. So here, these infrared display technology, so these antibodies are cloned under the P3rd gene of these phage meat, and they are displayed on the surface of the phage. That's why we call them as a phage display technology. And this technology was developed by Professor Smith and Winter, and they have been awarded the Nobel Prize in Chemistry in 2018. And one of the major advantage of this technology is that roams around bacteria and bacteriophages, and we know it's easy to culture bacteria and bacteriophages, especially in the resource limited conditions. And these antibody can be libraries can be used against wide variety of antigens. And if we have these naive library, so they does not require any immunization, and they can be these antibodies can be engineered in different way. We have a freedom to operate on working of engineering of these antibodies. So mostly these libraries are of two different origin. If we make from the healthy donors, see we mostly capture the IgM reporter, and if we make from the immunized animals or vaccinated individuals or the recovered individuals, so we mostly cover the IgG reporter. So these are mostly known as immune libraries. There are other parts also we call semi-synthetic and the synthetic libraries. So, in my work during translational health science and technology during the COVID pandemic, so we started working on the SARS-CoV-2 virus. So we know that RBD receptor binding domain was one of the major target of the neutralizing antibodies. So we at that time express RBD protein in the mammalian expression system so that we have a native conformation of this antibody. So then I got the opportunity to work on the Tomlinson ING library that was developed by Professor Winter. So these libraries were available with our collaborators. So at that time, we screened those libraries with the RBD protein, and we were able to isolate a good number of binders against these RBD proteins. So these single chain antibodies were then tested for their neutralization, and these antibodies were able to neutralize the SARS-CoV-2 viruses. And then we found that these antibody mostly binds with some of the conformational epitope in the receptor binding domain. And then we converted them into three different forms: single chain antibody, scFv, Fc, and IgG1. And then we tested for their binding affinity. So these nanobodies were binding with the nanomolar affinity. So all these works we were able to finish in 4 weeks time. So this was the first paper where we have shown that these naive antibody libraries can be potentially used for developing these antibodies against the SARS-CoV-2. And JBC was generous enough to accept this manuscript at that time. And then we also developed some of the platform where we found that some of these antibody clones contain the stop codon. So we developed a strategy where we can express these antibodies without removing the stop codons and by changing the bacterial expression system. And we developed a rapid strategy where we were able to screen these antibodies that contains the inherent amber stop codons. And then we establish a protocols where we can screen these libraries in a very high throughput way. And next, we were interested to see whether when we increase the valency of these antibodies from bivalent to tetravalent, whether they are able to increase the neutralization potential. But in our case, we found that there were not much change in the neutralization potential. So it confirmed that even the bivalent antibodies were sufficient to neutralize these viruses. So but at that time, a number of companies approached us and they were interested to license these antibodies. So we wrote email to Professor Winter that these libraries were originally generated by you, and we want to commercialize or license these antibodies, then Professor Winter mentioned that these libraries are actually only for the research use, not for the commercialization purpose. And then in the last, he wrote to me that Rajesh, is better to make your own library. So at that time, it was not possible to make own library. So then we went back to our traditional technology that is mostly known as the hybridoma technology. And then at that time there was emergent of alpha, beta, and gamma variant, and we found that there was a mutation in all these three variants, that was N501Y mutation. And then later these delta and omicron came when we started our study, and we found in omicron also we found N501Y mutation. So we thought this mutation can play an important role. So we recombinantly expressed this protein, and then we went back to the our traditional technology that was developed by Collard and Meistein, that the hybridoma technology, where we can fuse the B cells with the myeloma cell and we can generate the hybridoma. So we immunize the animals with the RBD and 501Y protein. And after the second boost, we tested for their binding, and we found that all the animals develop a high titer of binding antibodies. And when we were mainly interested about the neutralization profile, and then we tested these animals, we found that all the four animals were able to neutralize at that time the variant that were prevent SARS-CoV-2, wild type, alpha, beta, gamma, kappa, and delta. So we selected animal 3 at that time because there was a high titer of neutralizing antibodies in that animal. So we sacrificed that animal and we made the hybridoma, and we were able to make around 40 monoclonal antibodies that were binding to the receptor binding domain. So out of these 40 antibodies, when we tested for the neutralization, so these four monoclonal antibodies were able to neutralize the Wuhan Kappa, Delta, Alpha, and Beta variant that was present at that time, and we found all these four are broadly neutralizing antibodies. But when the Omicron variant later came, when we tested, we found that out of four, three were able to neutralize, and this P4A2 antibody was able to neutralize all the SARS-CoV-2 variant at that time. And then we further characterize this antibody, means what are the mechanistic, how this antibody is neutralizing, what are the epitopes, conjure epitope that are present in all the SARS-CoV-2 variants. So we tested for the neutralization, and we found that this antibody was equally efficient both against the Delta and the Omicron variant. We performed immunocytochemistry experiment, and we proved that this antibody binds with the viral infected cells both at the Omicron and the Delta, and we were interested to look for the affinity. So we took this antibody, and at that time, Sortimab was the FDA approved antibody, and we compared the affinity, and we found that both of these antibodies has a equal affinity, 1.7 nanomoles and 2.09 nanomoles. And then we tested for the neutralization potential, and we found that both the antibodies were able to neutralize, but P4A2 antibody was slightly better than the Sortimab that was able to neutralize both Delta and the Omicron variant. So then we compared the data with the other approved antibodies at that time, and we found that this P4A2 was able to neutralize very well efficiently as compared to the other FDA approved antibodies that were available at that time. So we were interested to see, means what are the mechanistic view, how this antibody is neutralizing all the variants and how it works in the animal models. So first we look at the epitope, and then we performed a competitive BLI experiment where we did a competition of the antibody with the AC2 binding site, and we found that this antibody was competing with the AC2 bind. So it confirmed that it binds with the receptor binding domain where exactly the virus binds with the AC2 receptor. So that confirmed that this bind with the where the virus binds, and it blocks the virus directly entry. So next we were interested to see how this antibody binds with the other variants, and we did a simple ELISA experiments, and we found that this antibody binds with all the variants that were present at that time with a binding profile that give a good EC50 value. And then we look for the affinity determination. So and we found that this antibody binds with nanomolar to picomolar affinity with most of the variants that were present. Here you can see the BLI graph that clearly shows that these antibody binds with the nanomolar to picomolar affinity with all the SARS-CoV-2 variants. So then we perform the immunofluorescence staining, and you can see the antibody was able to bind with all the variants with equal foci count experiments, and it further proved that this antibody also binds with the viral infected cells. So next we were interested to see whether this antibody is able to neutralize the SARS-CoV-2 variants or not. So we took AC2 transgenic mice and we prophylactic given these antibodies 12 hours before or 12 to 20 hours, 4 hours before, and then we infected with the virus. And then we look for the different parameters like body weight and the viral titer. And here in the graph you can clearly see the animals that were given the antibody prophylactic, we were able to see a good survival and the weight rate, and there was a reduction in the viral titer also. So this confirmed that this antibody is working well when given prophylactically. So, but we know in the hospitals what happens, most of these patients comes with the symptoms and they have some or other problems. So we were interested to see what happens when these antibodies are given therapeutically. So we have given the virus and then after 12 to 24 hours, these antibodies were given to the animals, and then we selected the kappa, delta, beta and other variants, and we found that in all the cases, the antibody were very effective in case of the therapeutic interventions, and especially when they are given in the low dose around 5 milligram per kg of the body weight, and there was a significant reduction in the viral titers. And you can see the lung pathology also, and you can see that there was a drastic improvement in the lung. And then we were interested to see whether the antibody is binding with the SARS-CoV-2 and is neutralizing the SARS-CoV-2. So whether it is effective against the other alpha and beta coronaviruses. So we performed the pseudovirus-based neutralization assays, and we included SARS-CoV-1, MERS, and the other common cold coronaviruses. And we found that this antibody specifically binds with the SARS-CoV-2 and does not neutralize any of the other alpha and beta coronaviruses. So this confirmed that the epitopes are specific and present in the SARS-CoV-2 variants only. So next, we were interested why this antibody is so effective against most of the variants at that time that were circulating. So then we performed the X-ray crystallography of these antibody in collaboration with from Regional Center of Biotechnology, Professor Deepak Nair, and we solved the crystal structure of this antibody, and we found that this antibody binds exactly on the same epitopes that are very crucial for binding of the virus to the ACE2 receptor. So this confirmed that this antibody exactly mimics the binding site of the ACE2 binding site. So conclusion from this part of our study was that the binding point of P4A2 antibody on the RBD differs from the epitope of the previously known class antibody, and it binds on the same site where AC2 is binding. And we found that most of the variants this antibody was able to neutralize. And then one of the major limitations of our study was that these antibodies were developed in mice, and because we cannot use these antibodies directly for the clinical testing, so limitation was that we have to humanize this monoclonal antibody. So when we were working on the humanization of this antibody at that time, we know the other variants of the SARS-CoV-2 came, and when we tested this antibody, so this antibody was not effective against the Omicron variant BA.5. So means it was working against all the variants, but when BA.5 came, so this antibody was not able to neutralize. Then we thought one option is that we have to make this again new antibody and look for which of these antibodies are effective against the BA.5. So we thought this option will be a very tiring option, go back and then immunize the animal and get again the antibody. So we having the structure of this antibody, and we designed the humanized template of this antibody, and we generated some degenerate primers, and we constructed a far display library of this semi-synthetic where we introduced the random mutations, and when we screens against the BA5 variants, we were able to get some of the antibodies that were very well binding both with the BA.1 and BA.5. And we've got some of the monoclonal antibodies that were now able to neutralize both BA.1 and BA.5 variants of the SARS-CoV-2. So we proved that if we are able to make these antibody engineered by using these fast display approaches, so we can save a lot of time to engineer these antibodies quickly. So summary is that structure guided phage display based library approach is a powerful tool that can generate high affinity binders against the escape mutant in a limited time frame. So this was one of the study that I worked when I was a scientist in the THSCA Faridabad. So after moving to my new institution, IIT Roorkee, so I was remembering the words of the Professor Winter that you have to make your own antibody library because every time it's not possible to make mouse antibodies and then humanize those antibodies. So after joining here in IIT Roorkee, so my lab started working on making human new far display antibody libraries, and then we also started working on the nanobody because these camel and shark contains unique class of antibodies that lack the light chain domains. So they contains only the heavy chain antibodies and they are able to bind with the antigen. And these heavy chain antibodies are approximately 15 kilodalton. So that is approximately 10 times smaller than the human antibodies, and they are high affinity. And because both of these animals live in the adverse conditions. So these and single domain antibodies are very highly thermostable. They have a good solubility, and there are also reports that these antibody can, if engineered, they can also cross the blood-brain barrier, and they are devoid of the FC region of these antibodies. So they have a high tissue distribution rate. They can go and bind with the cryptic epitope. So we collaborated with the Central Marine and Fisheries Research Institute in India, and we thought we will start with making the nanobodies from the shark animal. And here you can see one of the major advantage of these nanobodies is that they are very small in size. They can bind, and the binding pockets are different as from the human antibodies. So maybe some of those antigens that are not recognized by these human antibodies, possibility is that these shark nanobodies can recognize those epitopes that are specifically cryptic in nature. So, keeping all those rationales in mind, so we thought we will construct a large neo mega diversity, the VNR shark nanobody libraries. And the second goal was that how we can make a high throughput screening platform so that we can isolate these nanobodies in a high robust way. So, starting with one of the first work, we collected these bamboo shark from our Indian Ocean, and then we collected the spleen sample. Spleens are the rich source of these B cells. So we collected the B cells and then we isolated the RNA and we prepared the cDNA. And then we went back to the genome of these bamboo shark and we performed the NGS sequencing, and then we designed some of these unique primers that were able to amplify these VNR antibodies from these shark. And then we cloned these into the phage mid vector, and we constructed a phage library of 10 to the power 11 clones, and then we did some of these functional validation. So one of the major concern was that we know in far display, if you have a good library size, chances are that you will get a good number of antibodies after screening against the antigen. So we developed this antibody library when we determine the size of this library. So we found that this library size is around 1 into 10 to the power 11. So next question comes when you make this library, whether insert size of your clones in the library is perfect or not. How many clones in your library has a proper insert size? So then we performed a simple colony PCR analysis and we found that almost 97% of the clones in our library has a positive insert. So next we were interested and performed a simple restriction analysis of some of the randomly selected clones, and we found that similar results we got, and 98% of the clones were positive in our library. So next question comes, you have a proper insert size, 98% of the clones has a insert. So whether those clones are functional or not. So we performed a simple Western blot analysis and we found that 85% of the clones in our library was functional. So at that time we were confident we have developed a good library size of 10 raised to the power 11. They have a proper insert size confirmed by the colony PCR and restriction digestion. And these nanobodies are functional in stage also. So we had a confidence now this we can proceed more. But all of these experiments done only on a limited number of clones, maybe less than 100 clones. So, but library's diversity was 10 raised to the power 11. So that does not represent the overall diversity. So then we went back and we performed the next generation sequencing analysis. So we amplified our library and then we sent it for the NGS sequencing. So when we sent, so here you can see zero point 0.3 million VNR sequences we got after the NGS sequences. First, we look for the number of the cysteine bonds because these cysteine bonds are more frequently present in these sharp nanobodies, and we found that most of these nanobodies contain at least one cysteine bond, and around 30 to 40% does not contain any cysteine bonds, and around 12% contains two cysteine bonds, and three cysteine bonds were present only less than 1% of the clones. Then we looked for the CDR3 diversity, and we found that there was a diversity ranging from 4 to 24 clones, but average length was found to be 12 to 13 amino acid. That was as expected, as reported by in other groups also. So when we look at the sharp nanobody, there are four different types: type 1, type 2, type 3, and type 4. And these type 4 are towards more similar to the human antibody gene sequences in case when we compared for their humanization potential, how we can humanize these antibodies. So these type 4 are very close, and there are reports that confirm that these type 4 antibodies can be easily humanized. So we were interested to see how many type 4 VNR nanobodies are in our library. So we look back to the literature and we found that in most of the literature, type 4 VNRs are only 1%, or in some of the cases, they were even less than 1%. But when we look at the data and when we read those papers, we found that most of these information was based on the immune library. There were no information available on the name library. And we found that in our library, 40% of the clones, 35 to 40% of the clones were around type 4, and these type 4s are structurally very flexible and they can bind to the cryptic epitope. So when we look back to the structural data, so there was no structure available for the type 4 VNR nanobodies in the literature. So we were interested how these type 4 look like, whether they are different from the type 1, 2, 3 VNR, or how close and similar they are to the human antibodies, and how they are close or similar to the cameloid nanobody. So we took two nanobody, one having a moderate CDR and one having a short CDR. We recombinantly expressed these nanobodies in our lab, and we found these nanobodies has a good expression profile in our lab. And then we took these two nanobodies for the structural analysis, and we performed in collaboration with our department with Professor Pravinder Kumar, and he helped us in solving the crystal structure of these two nanobodies. And we were lucky that we selected two clones, and both two we were able to get and solve the crystal structure. And as you can see, both of these nanobodies have a different CDR, and you can see the flexibility. When we did the structural modeling, and you can see some of the clones that we obtained from our library, and you can see our library clones has a structural variability in the CDR3 region that also again confirms that our library clones have a good structural flexibility in the CDR3 region. So now we were very sure we have done a number of parameters that confirm that this library has a good number of PNR clones. They are diverse based on their sequences also. Structural flexibility is also there, and they are functionally making the good nanobodies amount in the in vitro expression system. So then we thought, means whether we are able to isolate the binders from our library or not, because that was one of the biggest challenge. Because naive libraries are known that getting a good binding antibodies are difficult, but report says that if you have a good library with a diversity, there are possibility you can get a good binders. So we confirm all the parameters in our study that all the things are right. So now we move back to the screening of the antigen. So to prove whether these naive libraries are able to make antibodies or nanobodies against the diverse antigen, we selected two viral antigens, RBD protein from the SARS-CoV-2 and alpha protein from the Japanese encephalitis virus. We selected two cancer antigens, breast cancer and lung cancer. We selected one autoimmune antigen. We selected some of those plant proteins. We selected toxins from the snake venom. We selected proteins from the alga origin and proteins from the parasite origin. So we almost covered antigens from most of the important uh, antigens that are in the different phylum of these organisms. So we started with SARS-CoV-2 and we screened our library against this RBD. And after three rounds of biopanning, we were able to get around 10 clones. And when we sent these for sequencing, we found three clones were unique. And out of all the three clones were from the type 4 VNRs. So we were able to get a good binding in the phage ELISA. So the next question comes whether these antibodies are binding with the antigen with a good affinity, because in naive library one of the major challenges that you will not get a very good binders. So we performed our BLI experiments and we showed that this nanobody is binding around 2.87 nanomolar with the RBD antigen. So next, we selected envelope protein of Japanese encephalitis virus. We performed the similar exercise, but in this case, we out of 16, 12 clones were unique, and few of the clones were type 4, and some of these clones were type 2. We performed again the BLI experiment, and this nanobody was showing 53 nanomolar affinities. Next, we selected the SORT1 antigen from the breast cancer. We screened against, and here we were able to get almost 100% diverse clones, and one of the antibodies or nanobodies were binding with this antigen with the picomolar affinity. So out of these 8, we got one clone that was binding with the picomolar affinity. So next we move back to the lung cancer antigen, and then we selected plant proteins. We performed the similar experiments. We got antibodies, nanobodies of type 2 and type 4, and unique clones were about 85%, and they were also binding with the 11 nanomolar affinity. Then we selected one of the parasite proteins. Again, we did the experiment, and all type 1, 4, and type 2 antibodies were there. Uniqueness was 81%, and they were binding with 33 nanomolar. Then we selected venom from the cobra and we did the biopenning and we found that and nanobodies were binding with the nanomolar affinity with the cobra venom also. We selected one of the well-characterized antigen BEF and we were able to make nanobodies against that also and they were binding with the 19 nanomolar affinity. We selected against the algal protein and then this is the summary table that you can see we selected around 9 antigens and the hit rate was almost 100%. These are the number of clones that we sent for sequencing and these are the number of the unique clones and you can see the minimum number of unique clones were 30% and maximum we were able to get 100% unique clones and affinity ranges especially in case of nanomolar and in some of the targets we were able to get the picomolar affinity also. So next we improvise our screening strategy because when we screen these antibody libraries, so how much time it takes, we optimize our screening strategy and in 14 days time, we were able to complete the whole biopanning process and able to get some good binders in the soluble ELISA also. And the hit rate of our antigens, still now we have screened against around 15 antigens and it's approximately almost 100%. So conclusion from this study is that we have constructed a large VNR nanobody library from the bamboo shark that has a diversity of 10 to the power 11, highly diverse. It covered different classical functional VNR, type 1, type 2, type 3, and type 4. And type 4 are more important, especially for the humanization purpose. And this library contains around 30 to 40% of type 4 VNR. Until now, hit rate is approximately 100%. A number of the unique clones was 30 to 100%. And biopanning strategy, we have to optimize from antigen to antigen. But in most of the cases, after round 3, we were able to get a good number of binders. So the initial part of the work was done in translational health science and technology, the COVID part. So I want to thank all my collaborators from Translational Health Science and Technology Institute, Regional Center for Biotechnology, my collaborators from AIMS New Delhi, and from National Institute of Immunology. And this shark nanobody work was done in IIT Roorkee. So I will acknowledge my collaborator, Dr. V. Santosh, who helped in providing the shark spleen samples, and my other collaborator who are working on different cancers. So they shared those antigens, Dr. Arun Kumar and Dr. Parvinder Kumar for doing the structural analysis, and my collaborator Dilip. And I will thanks LifeArc for funding this work and establishing this pandemic preparedness initiative, especially for the antibody work. So we did this work from the funding from the LifeArc, and we have different funding sources from ICMR, Ignite Life Science Foundation, Department of Biotechnology, IIT Roorkee funding, and ANRF funding. And this is my team in IIT Roorkee that has worked on these VNR antibody libraries. And we have also established a spin-off company in Tides IIT Roorkee, where we are giving the nanobody screening services. And we are also working with some of those industries who are interested to make these nanobodies and take for the diagnostic and therapeutic applications. So we have a startup here. So thank you all for listening and your time. So I have finished maybe my talk before time, so I will be more than happy to take question and will engage in the discussion part.
Thank you so much, Dr. Kumar. Go ahead and navigate to the bottom of your screen to the Q&A section, and that will allow you to write out your questions, and I will read them aloud. But while people are writing them in, I actually have a couple of questions to start. And it's sort of a two-in-one. So before you're able to start building up your library, so say there's a new virus, we'll just call it virus X. A new virus is out there, it's causing disease, some sort of outbreak has happened. What information do you need in order to begin to build your library?
So actually, we actually don't have to start to make the new library because these are the new library as we have shown against number of antigen that a single library can be universally used to isolate these nanobodies. So one of the major information is that if we know what are the envelope proteins of that virus that are responsible for binding of the virus to the host cell, and if we have some information about the structural epitope or the fusion epitopes or the binding epitope, so that will be very informative for us to plan our screening strategy and make these nanobodies.
Awesome. And so on that, your 14-day time period to start creating these nano antibodies, is that universal as well, where it doesn't matter what you're targeting, it's still that 14-day time period?
Yes.
Awesome.
So yeah, we have tried against multiple antigens, and till now means we have got approximately 100% hit rates. And but one of the limitation is that when you make a immune library or get antibodies from a immune recovered individuals, definitely there are chances that you have multiple options of the affinity matured antibodies. But although those options are not available in the naive library, but our affinity data shows that most of these antibodies are in the nanomolar affinity range. So that can be means a good start to make these antibodies and use especially for these pandemic preparedness initiatives.
Thank you. So going over to our chat questions, first one, what is the significance of looking at cysteine residues in the CDR3 region of the nanobodies, as in why only cysteine?
Yeah, because these shark mostly lives in the adverse condition and they produce more urea so that they can tolerate those salt conditions. So these antibodies contains more cysteine. If there are more cysteine, so there are problems in the humanization of those antibodies, maintaining their conformation. So type 4 VNR only contains one pair of cysteine. So it's easy to humanize all those cysteines, and when we use for the therapeutic application, there are chances that these cysteines can interact with other autoantigens also in the human body. So less number of cysteines are more preferred when we are humanizing these antibodies.
Okay. And so the second part is there were a lot of type 2 VNRs that were also seen abundantly alongside type 4. Is it possible to use those type 2s to be optimized for humans?
Yes, yes, there are possibilities, and we are also trying to make some AI-based pipelines where we can, because now we have a millions of sequences from these VNR after the NGS. So we are trying to develop a AI-based platform where we can humanize these type 2 VNR also, because there is no database available for these VNRs that can be given out the train. So in one of our project, we are going to train these AI-based humanization platform for VNR. So let's see, maybe in next couple of years we have a data whether these type 2 and type 4 how they work, especially when they are humanized.
Awesome. Okay. We have another question here. How do your platforms address the risk of developability issues? How low expression yield early in the screening phase?
Yeah, actually we have worked on both nanobodies from camel and shark. So in shark, we have found that these nanobody express in very good quantity, because when you do the X-ray crystallography, you need a lot of protein. So we performed the crystal structure of these nanobodies, and we found that most of these clones were expressing very good. But irrespective, when we went to the camel VHS nanobodies, so the expression level was a big problem for us. So we more focus on these VNR antibody, and they have a very good high titer of expression level, especially in the bacterial system, and we have optimized all those parameters also. And other thing they have asked about the ADE, because these nanobodies does not contain the FC, so then there is very minimal chance of having these ADG issues, because these are devoid of the FC region, so they can
Okay.
go and directly bind with the antigens.
Okay. And so the final part of this question, how do you validate that your universal library maintains functional structural diversity against complex, conformationally dynamic viral trimeric spikes rather than just recognizing unfolding or recombinant monomeric target antigens?
So actually, some of the data I have not presented here. So we have used Nipah virus F proteins that are stabilized by the cysteine bonds, and we were able to make some neutralizing nanobodies that are able to block the entry of the Nipah virus into the host cell. So we have tried against those complex antigens also that makes the trimer, because Nipah F proteins make the trimer, and we have used those trimers because in such cases we mostly coat, or we use the nickel coated plates, and then we add the his coated antigen, so they does not flat those antigens on the ELISA plates, and they have some conformation. Or if we are using the FC based antigen, so we are using the anti FC receptors on those plates so that the antigen in a proper conformation. So we are trying to optimize those assay, and we have got a success and using these. So this library can address those issues also.
Thank you so much for that. Do we have any last minute questions? Go ahead and type them out. Okay, so in the absence of any additional questions, I just want to thank our speaker again and let everyone know for our June, we will not be having a webinar, but for July, our next webinar is on the 30th of July, and we will be hearing about HIV-1 and neurodevelopment. Thank you again, Dr. Kumar, and I hope everyone has a fantastic day.
Yeah. Thank you. Yeah. Thank you. Thank you.